Hello, everybody, and welcome to the ACCRF's fifth patient webinar on the latest research update in adenoid cystic carcinoma. My name is Dr. Božo Todorić. I'm a retina surgeon and sit on the board of directors at ACCRF. On behalf of everybody at the foundation, we're so happy to have you join us today as we share the latest news in ACC research. Since its inception, the mission of Adenoid Cystic Carcinoma Research Foundation has been driven by efforts to find better treatments for ACC and ultimately a cure for this disease. We're strong believers in reasonable hope that drives our efforts towards this mission. We always want to ensure that all of our pursuits of this mission are grounded in hard science and clinically repeatable results that can reliably benefit patients. This is why for nearly 20 years, we have been dedicated to building a global network of oncologists, basic and clinical and translational researchers, and supporting their cutting-edge research that's considerably advanced our understanding of ACC and its treatments. Now, more than ever, we have better understanding of the mechanisms that drive ACC cancer proliferation progression. There are specific biological targets that may be able to slow and stop disease. We know that alternation in, for example, a gene called MIB is a defining characteristic of ACC. But besides MIB, there's number of other biological targets that can be used to develop specific cancer therapeutics. As Jeff and Nikki will discuss in more detail shortly, the most recent preclinical data on these MIB inhibitors and antibody drug conjugates in preclinical models are most impressive to date. And these results are setting a very strong stage for clinical trials in 2024 and 2025, and these are shaping up to be most exciting and promising yet. While there's still much work ahead, we're heartened by the headway that's being made every day as we speak. But most of all, we're incredibly grateful to all the brilliant, dedicated physician scientists who continue to drive this research forward, which gives us all hope, and to you members of our patient community who remain the heart of our organization. You're the main and only reason why ACCRF is doing this, and without your support, our efforts would not be nearly as efficient or effective in pursuit of this mission, which is so deeply personal to all of us. Therefore, with sincerest appreciation and gratitude, we thank you for your ongoing support and partnership. And now I'd like to turn things over to co-founder and executive director, Jeff Kaufman, also known as the Man, and our director of research, Spardy Burr, who's going to take us to the main part of the presentation.
Hello, everyone. My name is Jeff Kaufman. I'm the executive director of ACCRF, and I want to say thanks so much to Božo. It's absolutely been wonderful to have an MD, a PhD, and such an energetic driver of research on our board of directors. Božo has done an amazing job of highlighting areas for us to focus on, and we're so appreciative of all his help. So now let's go to today's agenda. Today, I'm going to spend a bit of time we had Božo's talk, which we're thrilled with. Now I'm going to spend a bit of time describing ACCRF's activities and the outlook for systemic therapies. Then ACCRF's Director of Research, Spardy Burr, will provide an overview of this summer's ACCRF Research Conference, as well as an exciting new type of cancer drug called an ADC, or antibody drug conjugate. Then we'll bring in Reed Moats, who joined ACCRF earlier this year to guide our communications and development efforts. Reed will be the moderator for the questions that we've received from you, from the patients, and he will wrap up the seminar. Before we get going with the agenda, let me go over a few housekeeping issues. The first is that the questions after the presentations were drawn from the webinar registration forms. However, we will also be monitoring the chat for additional questions of broad interest if time permits. So feel free to put some questions in the chats. We may or may not get to all of them. If we don't get to your question, feel free to email us at info@accrf.org, and we'll try to address them later. Secondly, please do not put any personal information, such as your medical history, into the chat or publicly disseminate any information from the chat. ACCRF does not provide medical advice. We seek to educate patients so they may discuss treatment decisions with an experienced physician in a private setting. Please don't make major decisions based only on this webinar. Make sure that you're talking to your doctors. And lastly, this webinar is being recorded and will be posted on the ACCRF website. It may take several days to get up there, but eventually it will. Oops. Now, as we start to discuss ACC research, it's incredibly important to put it into context. This webinar will focus on treatments for ACC patients with advanced disease. That means patients who have widespread and growing local recurrences and or distant metastases. The fact is that about half of all ACC patients are cured by surgery and or radiation, and they won't ever be at risk of developing advanced disease. We don't know who that half of patients are ahead of time, so it's important that all ACC patients are monitored for decades or for life to make sure there are no local recurrences or distant metastases. Please remember that there are many different patient journeys, so don't assume the worst. You may never develop metastases, and even if you do, they may be manageable for many years or decades. That is what we at ACCRF are working on every day. So who are we? This is not okay. ACCRF is a public charity with a mission to accelerate the development of improved therapies and eventually a cure for ACC. My wife Marnie and I started ACCRF after she was diagnosed when we had 4 boys under the age of 10. Just like you, we desperately want better treatments. Looking at our leadership, ACCRF's board of directors is made up entirely of ACC patients or their close family members. So we understand the urgency to make progress. And our scientific advisory board is made up of world-leading oncologists who steer our research in the most promising directions. ACCRF's strategy is to develop a pipeline of clinical trials based on the best available science that clearly demonstrates benefits for ACC patients. And at ACCRF, we're very proud of our accomplishments over the past 18 years. We've awarded more than $20 million in funding to our research network that includes about 50 grantees across 30 institutions in the Americas, Europe, and Asia. Our grantees carried out the genomic studies that identified the MYB gene translocation that characterizes ACC, as well as the Notch gene mutation that often drives more aggressive cases. Almost unique among cancer research foundations, at ACCRF, we manage our own set of ACC mouse models that we make available to all academics and companies. We've forged collaborations with more than 30 biopharmaceutical companies, bringing in their drugs to test on our preclinical screening platform, with the most promising drugs often going into clinical trials. And now we've got more than 20 clinical trials that are open, far higher than years ago when we first tried to attract biopharma interest to our rare cancer. But the most important accomplishment relates to patients and are educating you about past, present, and future treatment options. Now let me turn to a summary of the outlook for systemic therapies for ACC patients with advanced disease. Now remember, many patients don't have advanced disease, and these will be less relevant to you, certainly at the moment. When I say systemic therapy, I mean drugs that are either infused or in pill form, and which travel through the entire body system. Systemic therapy includes chemotherapy, targeted drugs, and immunotherapy. Surgery and radiation are usually called local therapies because they directly affect one area of the body. while drugs are body-wide or systemic. Systemic therapy is used only for patients with widespread recurrence or many distant metastases that are clearly growing. We've seen big advances in the use of systemic therapies in the past 2 to 3 years. Clinical guidelines for medical oncologists who treat ACC patients now mention VEGFR inhibitors such as lenvatinib and axitinib as reasonable treatment options for advanced ACC patients. They are not cures and they come with lots of side effects. But these VEGFR inhibitors do slow down metastases in many cases and may bring patients some valuable time. Nikki will share some data with you, but I will provide a small spoiler and say that the recent preclinical drug screening data is the strongest ever in 15 years we've been testing drugs. As these latest drugs are starting to enter the clinic, I'm incredibly excited to learn how patients do. Now, there's an important warning. What works well in a mouse does not always work well in people. So, despite our optimism, we continue to invest in supporting new drug development, making sure the pipeline of hope remains full for patients. My last point on this slide is that we've had recent clinical guideline updates, and we're hopeful that new drugs will change the way ACC patients are cared for in the next few years. So we feel it's an important time to ramp up and improve our education and communication efforts with patients. That's why we're thrilled to have Reed Moats join ACCRF to spearhead our communications. Reed has already made us much more organized and consistent in churning out email updates. And I hope you've noticed that in the last 9 months. We hope that there will be so many exciting developments to share in the next couple of years that Reed will be absolutely exhausted in the next few years. That's our hope. Lastly, I want to make sure that patients with advanced disease know where to look for clinical trials on our websites. Here at the top of the slide is the URL for our Open Studies webpage, along with some selected newer studies that we've added. In every case, ACCRF has helped bring the clinical trial to fruition, either by showing companies that their drug might be applicable to ACC based on target expression, or by conducting preclinical drug screens in our mouse models to prove the concept. Now let me stop here and turn things over to ACCRF Director of Research, Nicole Spardy Burr, to discuss our summer research conference in antibody drug conjugates. Nikki?
Can you see me okay?
Yes, you're coming
okay.
through.
Hello, everyone. I'm excited to start by sharing an update of the 2023 ACCRF Research Conference that took place in Boston, Massachusetts over the summer. We were fortunate to welcome 80 registrants from both academia and biopharma, most of whom are pictured here, with some folks coming from as far away as Australia to discuss recent advances in ACC biology and treatments. Next slide. The conference was held over 2 days and spanned basic science to translational and clinical research efforts. During the meeting, we heard about the development of three-dimensional ACC tumor cell cultures, which can be grown in a dish and are often used by companies or academics to screen a large number of early candidate drugs to determine which drug works the best to stop ACC cells from growing. From Dr. Renata Ferrarotto at MD Anderson Cancer Center, we heard preliminary findings about new biomarkers that may predict a patient's prognosis as well as sensitivity to certain drugs. We also heard her efforts to catalog the growth pattern of lung metastases in ACC patients. This work will provide crucial context to the FDA, medical oncologists, and companies when they're trying to understand whether a drug has a positive clinical benefit. From Dr. Alan Ho at Memorial Sloan Kettering, we learned about additional ways in which ACC tumors may hide from the immune system. This work provides important clues as to how we might reprogram the immune system to attack ACC cells with new therapies in the future. And in the next few slides, I'll share some very exciting preclinical data for new MIB inhibitors and antibody drug conjugates that was shared at the conference. Next slide. Before looking at the data, I'd like to introduce our PDX drug screening platform. Patient-derived xenograft models, or PDXs, are human tumor tissues that are engrafted onto mice. Our platform contains over 20 different models that were established from a variety of primary and metastatic ACC tumors. The PDX tumor tissue that grows looks like ACC, meaning that it has the appropriate tissue architecture, or often called histology, and they harbor gene alterations that are frequently observed in ACC tumors, like those involving the genes MIB or Notch. PDXs are useful to demonstrate whether a drug is active in a specific cancer. And when I say active, I mean, does treatment of the tumored animal with a drug cause the tumor to shrink or even melt away? PDXs can also help us to understand how normal cell processes become disrupted during cancer and to discover new drug targets in ACC tissue through things like sequencing. Because of these important activities, ACCRF has worked with over 40 drug companies to screen over 140 anti-cancer drugs in our models. And this preclinical work is usually one of the final steps before the FDA permits a clinical trial to proceed. Next slide. Work from numerous ACCRF grantees has shown that a protein called MIB is deregulated in the majority of ACC tumors and is likely required for the growth and survival of ACC cells, making it an extremely attractive drug target in ACC. Over the past several years, we've worked with multiple companies developing novel MIB inhibitors to explore their activity in our PDX models. An example of such a study is shown here. A PDX model that expresses high levels of the MIB protein is treated with either a control, so that's shown in the black line, or a novel MIB inhibitor at 2 doses. And tumor size is measured over time. You can see that the black line for the control group rises steadily over time, indicating that the tumor is growing fairly well. But excitingly, tumors treated with the MIB inhibitor either slow their growth massively, as is shown in the yellow line with dose level 1, or actually shrink away to nothing. And this is also referred to as a tumor regression, as is observed with the blue line at dose level 2. The picture on the right is showing an experiment that looks for MIB protein levels in tumor tissue, where any cell that has a significant level of MIB protein turns brown when you look under the microscope. We see that control-treated tumors stain very brown, indicating high levels of MIB, but that MIB inhibitor-treated tumors do not stain brown at all, indicating that levels of MIB are very reduced or gone. This experiment reinforces that the drug is doing an excellent job at hitting its target, MIB. The data generated with new MIB inhibitors represents an important step forward in shutting down one of the major pathways that ACC cells depend on for survival. We are very excited about this class of drugs. Next slide. On this slide, we're looking at a very similar type of PDX experiment, but that's now done with a different drug. So an antibody drug conjugate, or ADC, that targets a cell surface protein called B7H4. ACCRF worked with researchers at MD Anderson and AstraZeneca to identify PDX models that express high levels of B7H4 protein. You can see here that this specific PDX tissue stains very dark for B7H4, indicating high expression. We then tested this PDX model with several controls, so those are in the black, blue, and red lines, and two doses of a B7H4 ADC, which delivers a chemo only to cells that express B7H4 protein on its cell surface. With 1 dose of this drug at either level, we saw tumor regressions and in some cases a complete response, which means that the tumor shrank away to nothing and stayed that way for a certain amount of time during the experiment. Multiple companies are working to develop B7H4 ADCs. Each has a slightly different in the way that the ADC is made, how it becomes activated, or in the type of drug that's delivered to the cell. As Jeff showed earlier, a company called Seagen currently has a clinical trial testing their B7H4 ADC in ACC patients. Next slide. Okay, again, we're showing a very similar PDX experiment, but now testing an ADC that targets a different cell surface protein called AXL. ACCRF worked with researchers at UCSF and ADC Therapeutics to identify a PDX model with high AXL levels to then test either controls, in the black and the orange, or 2 doses of an AXL ADC in this AXL high PDX model. Either dose of the AXL ADC led to tumor regressions and in some case, a complete response. A company called BioAtla is currently testing their AXL ADC in a clinical trial that is open to ACC patients. I want to emphasize that the data I've shown here with the new MIB inhibitors and ADCs are some of the strongest preclinical data that we've ever generated. And I'm going to say what Jeff said, repeat what Jeff said in the beginning, that it's important to remember that how a drug works in a tumored mice does not always correlate perfectly to how a drug will work in humans. But this impressive data should fill us all with hope for the clinical trials that will test these drugs in ACC patients in the future. Next slide. Okay, so we've seen some exciting data on antibody drug conjugates, but what are they? So over the next few slides, I'll provide some context and explanation around one of the fastest growing drug classes in cancer. At its core, ADCs use a monoclonal antibody to deliver a chemotherapy drug to a tumor cell that expresses the antibody target. But let's break this down. And so we're going to start with, how does chemotherapy work? So within the body, there are normal cells, which we're showing here in light green, and cancer cells in dark green. Each cell surface is littered with proteins that send and receive signals, and within the cell lies the nucleus, which contains our DNA. Advance. When chemotherapy is given, it travels through the blood, and it's taken up by many of our actively growing cells, both cancer and normal, and causes so much disruption to the cell's DNA that they end up activating cell death pathways and are removed. Advance. On one hand, this is good because many cancer cells are destroyed. However, so are many healthy normal cells, which can cause unwanted side effects. Next slide. Okay, remember that I said that ADCs connect antibodies with chemotherapy. So the next step for our understanding is to dig into what are monoclonal antibodies. Monoclonal antibodies are part of our immune system that can recognize and bind to proteins within the cell. Binding of an antibody to a protein can trigger a variety of functions within the cell. But for cancer therapy, monoclonal antibodies are designed to bind and block specific cell surface proteins that are responsible for sending pro-survival signals to the cell. Please advance twice, Jeff. There we go. Blocking these receptor proteins shuts down the pro-survival pathway that the cell depends on and causes the cell to die. Advance. But what if not all the tumor cells express the specific pro-survival cell surface protein? Or what if some cancers don't have drugs developed for the pro-survival protein specifically expressed on their cell surface? Next slide. So that brings us to antibody drug conjugates, or ADCs. So this class of drug connects a monoclonal antibody to a chemotherapy drug using linker molecules. With this class of drug, you're no longer limited to choosing only cell surface proteins that send pro-survival signals, but rather we now have the flexibility to choose most any cell surface protein that's highly expressed on the cancer cell and not expressed or only very lowly expressed in a normal healthy cell. The monoclonal antibody portion of the ADC binds to the cell surface protein, and the entire ADC is internalized by the cell. Advance. Can you go back one more? The ADC is then broken down within the cell, and the chemo portion is delivered to the nucleus where it damages the DNA. Okay, advance. This leads to death of the cancer cells that express the protein target and sparing of normal healthy cells that don't express the target, which reduces unwanted side effects. Next slide. ADCs have a long track record of success in cancer, as supported by this table of FDA-approved ADCs targeting a variety of cell surface proteins in different cancers. ACC patients should feel very hopeful that there are many ADCs being explored in our rare disease. Next slide. So how do we find relevant protein targets in ACC? The foundation supports molecular characterization, so looking at RNA and protein of ACC tumors to identify and validate protein targets. An overview of one such project is shown here and was done by Dr. Renata Ferrotto at MD Anderson. Renata looked at the DNA, RNA, and protein levels across 54 ACC tumors, and from this analysis showed that there are two separate molecular subgroups within ACC. ACC1, which tends to have solid histology and is enriched for notch mutations and high MYC expression and have a poorer prognosis, express high levels of B7H4, while a second group, ACC2, which tends to have cribriform or tubular histology, high P63, less aggressive disease, expresses high levels of the protein called AXL. This work was fundamental in describing these two cell protein targets and motivated the preclinical work that I showed earlier, testing either B7H4 or AXL ADCs in their respectively high PDX models. ACCRF currently has multiple research projects dedicated to protein target discovery, and the goal of this work is to continue to better understand the target landscape in ACC tumors and to refill the clinical trial pipeline with promising new targets. Next slide. Okay, so some takeaways from the webinar so far. ACCRF's research conference demonstrated that many researchers are actively working on better treatments for ACC patients. The preclinical screening data that we showed for multiple novel MIB inhibitors and antibody drug conjugates is some of the strongest ever. Clinical trials of these promising drugs are just starting to open, with crucial data to be reported in the coming 2 years. ACCRF will keep ACC patients informed of new clinical trial developments and updated clinical practices. And we encourage everyone to sign up for our email newsletter as we communicate many of these changes through that format. Next slide. Now I'm going to turn it over to Reid Moats, ACCRF's Communication and Development Officer, to moderate the question and answer portion of the webinar.
Thanks, Nikki. Hi, everyone. I'm Reid Moats, and I will be moderating our Q&A today. It's great to have so many of you joining us. Thank you to everyone who submitted questions ahead of the webinar. I'm going to start us off with some of the more frequently asked questions that we received, and then we'll move on to questions that people have submitted via the chat. And a brief reminder to please not put any personal or medical information in the chat or to disseminate publicly any information that you see in the chat. And again, we are not able to provide any specific medical advice, and you should always consult with your doctors about any of the information that you see here today. If we're unable to get to your question today, please feel free to reach out to us on our website or via email at info@accrf.org, and we'll put all that information up at the very end so you don't have to remember it right now. But I think let's jump in. So to start off, I'd like to direct the first question to Jeff. Jeff, can you talk about sort of the typical prognosis for ACC?
Yeah, that's a very common and difficult question to answer, but let me try my best. Let me start by saying that the solid majority of ACC patients live for at least a decade from diagnosis. However, we know that there's a very wide range of patient outcomes and survival. You know, some patients, unfortunately, pass away within just a few years of diagnosis, while others live for many, many decades. Researchers have identified some characteristics of tumors that point toward worse prognosis. I think Nikki has mentioned some is solid histology, notch mutations. There's also 1p deletions, paraneural invasion, low P63 expression. However, even within these, the subsets Nikki was talking about, ACC1 with more aggressive and ACC2 with less aggressive typical growth patterns, there's huge variability. I mean, I know patients with notch mutations who are living very long and very active lives. So none of this is determined. Everything is sort of across a range. I'd also like to point out that the historical numbers are just that. They're historical. There are many brilliant researchers. We showed you some of their work already. They're pedaling very hard to make sure that the hopefully better treatments will mean that the historical statistics of the future, your outcomes, will improve in coming years. So, we got a wide range, but I wouldn't focus, only the median, the single median patient has the median outcome. Everybody else is different than that median. So keep that in mind.
Great, thanks, Jeff. And for our next question, Nikki, you talked about obviously a lot of clinical trials and some newer forms of treatment. Can you talk a little bit about what the best time is for a patient to be looking into clinical trials or other forms of treatments sort of in their treatment journey of ACC?
Yes. So I'll probably reiterate some of what Jeff led with, because I think these are important sort of take homes. So again, half of ACC patients may be cured by surgery and radiation and will never need to consider going on a clinical trial or some kind of systemic therapy. I think the issue is we don't know who those patients are, and that's why it's extremely important that all ACC patients, including those patients that have ACC at rare sites like breast or skin, they need to be followed, with regular scans at regular intervals. But broadly speaking, I think we typically think that surgery, radiation, or some other type of ablation might make sense if a patient has somewhere in the range of 1 to 3 metastases that are, larger than 1 centimeter. They don't really have smaller metastases, and that these larger mets are growing by at least maybe 20% over the past 6 months. Systemic therapy may be more appropriate if there are a larger number of metastases and that these are, at least some of these are actively growing or we call progressing. But I think it's important to reiterate that when to consider starting any type of therapy, including systemic therapy, which type of therapy should be an important conversation with your medical oncologist, and a patient should feel comfortable, seeking a second opinion when appropriate.
Great. And sort of keeping in the vein of treatment options, we've gotten a lot of questions both ahead of time and in the chat regarding chemotherapy and ACC. And Jeff, could you walk us through sort of chemotherapy as a treatment option for ACC, but also its potential efficacy in treating ACC?
Yeah. This is a frequent and important question. In my sense is that this is the topic that, is often misunderstood. So actually, I'm going to refer to a slide, if I can be technically capable. Where did I go? I'm sorry. Aha, here. Is that visible? Yeah.
It is.
Okay, super. So to start with, chemotherapy, is capable of curing a very few number of cancers. So there are some like testicular cancer and some childhood leukemias can be cured by chemotherapy. And that's a wonderful thing. The problem is almost all the other cancers are not fully curable by chemotherapy, and you might term them as chemo resistance. And that's where ACC is. We are chemo resistant, but that often in the patient community, I think, gets misconstrued as ineffective in all cases. And that's just not the case. Chemotherapy absolutely is not a cure. Maybe there's a patient or two that have been cured by it in the history of treating ACC patients, but it's certainly not a common thing. But that doesn't mean it's not effective in perhaps shrinking and slowing the growth and buying some time for ACC patients. Now we've seen some recent clinical trials of chemotherapy in ACC with response rates, which means major tumor shrinkage of over 30% in ACC. And that's kind of making us rethink, whether chemotherapy has some role in ACC. The older trials of chemotherapy were really from the 1980s and 1990s. They maybe had 8 or 15 patients, so these are very small studies. People were using X-rays, not CT scans, to measure the responses. And there was no standardized metric globally for measuring, partial responses. That didn't exist at that time. And so you had these very bad studies that really didn't tell us much. And that's partly why we don't know that much about how chemo really works in ACC. But we're starting to see these newer, better trials with better imaging, better, larger sizes, and they're showing some activity. So the other thing is, Nikki and I, we were trying to get our arms around this, and we sent out a survey to 20 medical oncologists globally that we thought really, really knew ACC well, and we got half of them responded, which is great. It would have been great if it was 100%, but half was great. And what we found was that 9 of those 10 people who responded, medical oncologists who really know ACC, they are using chemotherapy within their first, second, or third line treatments. Now, earlier treatments, the first line will often be a clinical trial or a VEGFR inhibitor like lenvatinib or axitinib. Those tend to be in the first and second lines, but there are some people treating with chemotherapy in the first line and the second line and or the third line. So this is widely used by people who know the disease. And the last point I'll make is that I know chemotherapy definitely, there are toxicity concerns. We know there can be side effects and they can be pretty bad. The one thing I'd note is that we don't, the other alternatives we have right now, like lenvatinib and axitinib, they have pretty rough side effects as well. They're a little different than chemo, but they're tough as well. So, and that's probably, we want to get past chemo. We want to get past the VEGFRs. We hope that in the next 2-3 years, we find really, really good therapies. But the reality is that, at least for a couple of years, patients are going to have to be cycling through multiple drugs. They may go on lenvatinib, then go on chemo, then maybe go on another drug. And that's sort of the way it is. So this is all to say we're rethinking the role of chemo in ACCRF. At our recent summer research conference, we did raise the possibility of whether we might support a new clinical trial to really get our arms around this and whether we can answer this question, very, very important question better than we have in the past.
Wonderful. Thanks, Jeff. And I'll direct our next question to Nikki. Nikki, can you first just explain a little bit about notch-related genetic alterations and then maybe talk about what options are available for patients who have those notch-related genetic alterations in terms of their ACC diagnosis?
Sure. So, we think that activating notch mutations, so that means that a mutation occurs in a portion of the notch gene, which basically kind of makes it super active. And notch is a gene that goes to the nucleus. And if you think of it like as a switchboard operator, it's turning on pathways in the cell that tell the cell to grow, survive. We think of it as an oncogene, right? So mutations in that gene that sort of turn it on, supercharge it, are going to occur in about 10% of patients with primary tumors, and then about 20-25% of recurrent metastatic patients. So I think it's important if you get your, tumor profiling document or whatever, and you see a notch mutation, I think the first step is to understand with your physician is the mutation in a region of the Notch gene that activates it. And they may need tests like the staining that I showed with the MIB and the AXL studies and things like that to kind of look at Notch staining in a patient's tumor tissue. So we think that these activating Notch mutations drive a particularly aggressive subset of the disease, which means that a patient may present with distant metastases even at the time of their initial diagnosis, or that recurrence is likely to happen much quicker. Notch mutant patients may develop slightly different disease course in the sense of their metastases may be occurring in places like bone and liver, which we see at a little less of a frequency rate in Notch wild type tumors or patients. So, going after the Notch pathway really makes sense for these folks. And if you go to our past clinical trials page, you'll see that single agent Notch inhibitors like brontektuzumab, AO101, and CB103. So these are all sort of getting at different steps within that Notch pathway and turning it off or down. They've all shown minimal signs of activity in Notch activated ACC patients, which means that, they've only significantly shrunk tumors in a smaller proportion of these patients. And in many cases, the tumor shrinkage was only was able to be sustained for a short amount of time. So the tumor acquired some resistance. So, from those initial studies, the thought is that combinations of drugs are likely to be necessary to make a meaningful impact on patient outcomes. So that means one drug turning off or down Notch and another drug turning off or turning down another pathway that's kind of feeding in and telling the cell to grow and divide. One such study that's currently open is combining the drug CB103 with lenvatinib, and that's currently open at the Dana-Farber. But we're aware of, new arms to this trial and additional new drugs that will make its way to clinical trials in the coming 1 to 2 years. I'd also like to point out that drugs outside of Notch inhibitors may also be relevant to Notch mutant patients. So, for example, as I discussed earlier in the webinar, Notch mutant tumors may express high levels of that cell surface target called B7H4, which are being targeted with ADCs. And so I think, again, this is a conversation to have with your physician, right? Notch and then other drugs beyond Notch, what are applicable to my tumor.
Thank you, Nikki. To switch gears a little bit, Jeff, are blood tests, or sometimes known as liquid biopsies, are those useful for ACC patients?
Yeah, this is another really rapidly growing area in cancer research. So these blood tests or liquid biopsies, are a way of measuring how active, how much tumor is in your body, and also whether they might have a Notch mutation or some other interesting characteristic. So these are really promising, exciting types of tests that are coming out there. Now, historically, if a patient presented to an oncologist, the oncologists would look to get a piece of tumor from the primary tumor, or perhaps a biopsy of a lung met, and send that tumor tissue to a tumor profiling company, which would then run a genetic profiling test to see if there were alterations that you could get a matched targeted therapy that the patient might do well on. And so historically, you always had to have that tumor tissue, and biopsies are not fun. So it wasn't, it could be hard to get that tissue, or the tissue was really old and wasn't as relevant to a patient's current most active tumors. So what the exciting thing about these blood tests, these liquid biopsies, is that first of all, it's just a blood draw, and it's quick, less painful, it's easier, it's cheaper. It's unlike just sampling one tumor tissue, you're actually getting information about all the potential metastases in a patient's body. So with a tumor tissue, you're just sort of taking one spot, but a patient might have 5 or 10 different metastases in their lungs, and you want to find out information on all of them. And you're more likely to get that from a blood test than sampling one of the tumors. So there are really some big advantages to these liquid biopsies. Now, it's early days in understanding these tests. There are some approved for other non-ACC tumor types. So you can get a liquid biopsy that then those results in other tumor types are actually leading to FDA approved, will point your oncologist to FDA approved drugs that you should try. That's the case for other tumor types. ACC right now is just for research. We've supported a few of these efforts. We want to understand, can you look for things in the blood of ACC patients? And it might be MIB expression, it might be methylation patterns. There are lots of different characteristics that can help us say this patient has ACC in their body or they don't, and what is it like? Eventually, I think this research, maybe on a 2-3 year horizon, we think that this will lead to better diagnoses of ACC. So you're maybe help with your primary diagnosis, but also figuring out, you may have had surgery on your primary tumor, gotten local control through surgery and radiation, and you have no evident disease in your body. But we all know that you have to monitor, and for recurrences and distant metastases, it may be that over time we can rely on these liquid biopsies to tell us if ACC has recurred, if it's come back. And the other thing that could be really useful is if it has come back, if it is there, it also could tell you which drugs might be most useful to attack that recurrence. So, summary, great, important area, just like new area like ADCs, still not quite ready for prime time in ACC, but we're hopefully getting there quickly.
Thanks, Jeff. And I think our last question to wrap things up, I'll direct towards Nikki. We've covered a lot about what the sort of current research and drug trial landscape looks like, but Nikki, can you give us an idea of what ACCRF's research priorities are and kind of will be moving forward into the future?
Sure. Jeff, I think we have a slide to kind of go over some new areas. Okay, so, I think we heard a lot today about cell surface markers and how excited we are about ADCs. But there are, other drugs or therapies that exploit cell surface markers that are currently being, researched and are in clinical trial testing. So things like CAR-Ts, radioligand therapy, a molecule called a BiTE, which engages the immune system. These are all in development. And so I think it's really important that the foundation expand and invalidate the molecular targets that are expressed on ACC tumor tissue. We have a variety of grants that are kind of trying to get at this from, using different assays or different technologies. And, really our hope there is that we see a lot of validation from one grant to another, where we're really honing in on, specific proteins that are expressed on ACC tumor tissue. And then in many cases, there are proteins that are expressed on a variety of tumors. So, like this B7H4 and AXL, these are therapies that were not developed specifically for ACC, but we can repurpose that. They're developed in other cancers, but they are also applicable to ACC. And so it's really important that we continue this discovery of what are these proteins. And I think, this is a really wide area of cell surface type therapies. And if this is an area of, going into CAR Ts and radioligand therapy, if this is of interest to the patient population and you'd like to see a webinar in the future that really covers these topics, please send us an email or put it in the chat, and we'll consider this as a topic for a future webinar. The foundation, needs to continue to facilitate biopharma entry into ACC. We do this by continuing to provide, easy access to our tissue microarrays to validate target expression and make it really simple, right, to access our preclinical screening models and look at whether their novel drugs have activity. We talked about the 3D cell cultures that ACCRF has been working alongside a company called Kiotech to develop. And our goal over the next year is to expand this platform. So that means bring in even more models there to see how they grow. And really our hope is that as we better understand this platform, it will mirror the success of the PDX models for earlier stage biotechs who are still trying to figure out which candidate molecule might make the most sense to move through their pipeline. And then finally, ACCRF wants to deepen our contacts with FDA and help generate relevant data on ACC natural history and clinical trial endpoints. Because as I mentioned, I think earlier, this is really crucial to help, the medical community really understand whether a novel drug is positively benefiting the patient.
Hey, thank you, Nikki. And I think that's a great place to end our Q&A. Thank you so much to Jeff and Nikki for all your really thoughtful and insightful answers and all the information you shared today. Thank you again to Bojo for joining us, for your comments, and also for putting so much great information in the chat. Before we end today, we do want to just quickly acknowledge and thank our many supporters, many of whom are with us today on the webinar. We have a few photos up here of some recent fundraisers that people have organized in support of ACCRF, including the annual nationwide Brave Like Gave 5K. We are deeply, deeply grateful to everyone who continues to work to ensure that ACCRF is able to advance the research you heard about today and to keep pushing us towards a cure. That work is not possible without the people like those that you see on your screen right now. And so we just wanted to acknowledge all of those wonderful people and say thank you, and also to say thank you to all of you for joining us. We're really grateful to have you with us today and to ensure that the ACC patient and advocate community remains strong and well informed. So thank you so much for being here. And then I think we have one last slide here. This is just a few ways that you can stay connected with us and stay informed and up to date about all the news around ACCRF. So we have our website there, accrf.org. We have our email address, info@accrf.org. Feel free to reach out about any questions that you might have had from this webinar, or just any questions generally you might have about ACC or the work that we're doing. You can email us directly there and we'll get back to you really quickly. And finally, if you haven't already done so, we would really encourage you to sign up for our newsletter, which you can do on our website and also through the link that Bojo provided in the chat there. Those go out quarterly, and those are just really great ways to stay up to date on everything ACC and ACCRF, including new research, events, and news that are late breaking. So we hope if you're not already a member of our mailing list that you'll join and stay in the loop. And I think that brings us to the end. So on behalf of everyone here at ACCRF, we want to thank you one more time for joining us today, and we hope to see you at our next webinar. We hope you enjoy the rest of your Saturday. Have a good day, everyone.